So thank you for giving me an opportunity to present this talk today on genome Jaunts. I want to share some excitement among children about what this is all about and why this kind of a topic. So this has its origin in a kid who joined my lab as a summer intern. So he wanted to spend some time in his summer vacation in my lab, and he joined as an intern. His name is Shreyas Maiti. And uh, this, what I'm going to share some excitement, which is also a part of this talk, is the this book which we have written, I along with another PhD student in our institute, Samita Matthew, we have written called the Genome Jaunts. Okay, and I would like to share this excitement why or how this book came about and why the interest in this book. And also, last summer was also very interesting because I had another girl join my lab. Her name is Jagriti. She is a 12th standard student now, and she came to my lab last year thinking what she wants to do in life and whether there could be something other than medicine and engineering, which she could also aim for. And some time spent in our lab made her think that she wants to take up a different career, and now she wants to be a neuroscientist. So how this came about is an interesting journey. She wrote in this Science Reporter. This year it got published, and I would encourage you people to pick it up, pick this issue. It's available online to read it, right? So coming to the book, or what made us write that book was actually a curiosity from this guy Shreyas. He is very interested in observing fishes. He has his own aquarium, and he knows lots of things about different fishes. But not only fishes, he's also interested in reading about different properties of different animals. For example, these are some things he jotted down for us, and for us to kind of give him certain kinds of answer whether we could as to why jellyfishes have such power of luminescence, where it uses it for attacking or predate, meaning attacking when a predator attacks it, then how does it actually use it to its advantage, and then it would switch off the light and exit the scene, right? Or something like this fugu fish. The name of the fugu fish is also called as puffer fish because when it feels very threatened, then it actually inflates itself, and then they also have a very poisonous sting. This fish is consumed in Japan as a delicacy, but if you are not able to cook this fish, then even people can die eating this fish. So there are some quaint features of this. Or he got me this information about these muskies, which actually to avoid predator, they form a group and move as if it's like a snake moving. Okay, and they are very difficult to catch, and that's why they also have got a name called fishes of 10,000 casts. I never knew about the existence of these animals before he came to me. And then he also talked about some sea turtle, which has an amazing biological clock or circadian clock, which allows them to lay their eggs at a particular time when the eggs would hatch, and the moon then again guides them back to the sea. So they are also vulnerable, and he keeps on worrying about all these animals. And one of the things which I asked him is that, would you want to know why they are so different, why you have such kind of animal with different kinds of behavior, and how do they come about like that, right? So I'll just spend a minute in asking you, or some of you can tell me whether you can read all the lines which I have written over here. If somebody can read out from the chat, if somebody has written an answer. Mm, it's a little kind of challenging to have a interactive one, but I am sure you people might not have been able to guess all the lines. Is it? Has anybody written in the chat box? Okay, we'll move ahead, but the what I wanted to kind of emphasize from here is that there is one common sentence which is shared by all of them, but they've been written in different languages, as a result of which we are not able to make out, right? If something is our native tongue, then we will be able to make out, and all of them actually read it as "we can do it." So the issue with why we could not read this was that everyone was written in a different language. Now imagine all all life on earth, all living organisms. For example, if we take bacteria, if we take plant, if we take chimpanzee, apes, human, the beautiful part about them is every life form is encoded in these four letters, which are four chemical letters, which are called as adenine, guanine, cytosine, thymine. So these are called as nucleic acids, and the order of this nucleic acid in a person will determine what that person is going to be, right? Meaning, so this is what leads to this thing which you would have heard about, DNA, the structure of which was elucidated by Watson and Crick. But you can imagine now, if every life form on Earth is written in the same language, then pretty much if you understand the language of these letters, then you might be able to make sense as to why this organism is different from the other. So I will just give an analogy that just like you have alphabets in English, which can be threaded to make words, and then that becomes a group of words can make a sentence with definite punctuation. And then if you put all the sentences together in a book, it will form a chapter, and a group of chapters will make a book, right? So similarly, in our cells, we have these nucleic acids which I mentioned, which if threaded together makes a DNA, which you can think of as a word, and then these DNA makes these sentences called genes, and these genes reside in the chromosome, and the entire chromosome is bound in something which is called as a genome. So as you have books in English or any other languages, the genome is what is called as the book of life, and the study of this book of life is what is the science of genomics. So just to recapitulate, you have the cells, and within the cells resides these chromosomes. So this is a human, so we have 23 pairs of chromosome, and in each of our tissues, which form the stomach or the intestine or lungs or whatever, we actually have cells which contain this genetic material, but some part of the genetic material is read in some part of the cell, and not everything is read in every part, as a result of which you get different organs, right? Now, what is very interesting is that these words or this DNA is not always. If you want to read it or you want to replicate it, it is not. Sometimes it can make errors. Just like if you are asked to write a sentence over and over again, then in the monotony you might end up making certain small errors. For example, you can drop a letter, you can insert a new letter, you can delete a letter, you can have another insertion, as a result of which, if you start with, let us say, one word like color, there can be a substitution which can lead to a word which doesn't make a difference. Then if it doesn't make a difference but it has to survive, then it can have an insertion but it makes a word which is totally different from what we started. Like that, through a series of changes, sometimes one word can assume a totally different meaning, based on how these small changes have happened, like the deletion, insertion, substitution, sometimes in inversion, sometimes a fusion. So you start with a word like color, and then you might end up with something like cool. So why I want to emphasize this is now you can put it in the analogy of a DNA. If the DNA we start all as same set of nucleic acid, then that DNA could also undergo different kinds of changes, as a result of which it can not only give differences to different members of a species, but it could also give rise to different kinds of animals, different kinds of species. Somewhere or the other, they are related to each other because through these same letters, as a result of which there could be some functions which are shared amongst all animals, and there could be something which can be unique for a particular animal depending on what kind of niche or what kind of adaptations they have. Now, how does this DNA convert to function? Because those are read as letters, but then the letter has to convey a meaning so that the cell can decide, now I have to do this, or this is the kind of function I have to have. So what happens is that this DNA, which is otherwise a double-stranded DNA, so there are pairing rules. So when you have a particular set of nucleotides, there is a rule of complementarity so that the other strand will make a complementary one. This allows it to replicate in a uh, without much errors. But what can happen is that this DNA can open up into a single strand, and then that is read through a method of what is called as transcription, which makes a mRNA. This is a message. So if you have a DNA of a particular nucleotide, in respect to the nucleotide present, it will actually insert another nucleotide through this process, and it will convert this whole thing into a kind of a message, and this message is called as a messenger RNA. So this happens in the nucleus, and then this messenger RNA moves out of the nucleus, and then the messenger RNA nucleotides are read in three groups of three. And what happens is these three are recognized as that if I have this message, then I have to have this amino acid kind of bound to this. And then if I have another three nucleotide which code for another amino acid, then the another machinery, which is called as a translational machinery, it actually starts reading this message and starts to incorporate amino acids. We have 20 different kinds of amino acid, and these particular amino acid will recognize a particular sequence, or which is called as a codon, and it will incorporate. So as a result of which these amino acids will start to get threaded, and this threaded amino acid is what is called as a what would form a protein. Okay, so length of amino acids which are read is called as a peptide, and when it folds into a structure which can perform a function, it would form a protein. So you can see that if there are changes in the DNA, then it will slightly change the messenger RNA, and if it changes this messenger RNA, another amino acid can get incorporated, because now it is read as a different message, as a result of which you can get a slightly different proteins. In some cases, for example, all of you must have heard about this disease called sickle cell anemia, where when we have these red blood cells, if you sometimes have a shape of the red blood cell which is a little different, and this compromises its oxygen carrying capacity. Now what happens, why does this happen, is because in the globin gene, which makes these cells, there is one change, as a result of which, if there is a glutamate, instead of the glutamate, there is another amino acid put, which is called valine. And the whole, just by this change, the shape of the cells can actually change to the sickle shape. So this is the consequence of a disease called sickle cell anemia. And that is why it is very important to know what is the normal sequence, and if people have certain kinds of diseases, maybe sequencing the part of the protein which codes for some functioning molecule, you might be able to know what is the change. So now the book of life is immense. It is like 3 into 10 to the power 9 base pairs. That means that many letters are there. And if a change can happen anywhere and we need to know its function, then we need to first know how many letters and in which order are they, right? So that led to a very huge project which was called as the Human Genome Project. It started in the late 80s. It took 20 years to actually sequence the first human genome. Now we can sequence a human genome in 3 days or in a day also sometimes. But this first one, to make a reference human genome sequence, there were a lot of technologies which were needed, and more than that, people started also having to, because if you just have the letters, it might not convey any meaning unless you know how to uncover the meaning out of that. Right. So human genome sequence was not just about knowing the letters, but also knowing their order and knowing the sentences so that you get to know what are the genes and then you get to know what are the functions of the genes. So this was a huge project, but because of this project, we actually got our first draft of the human genome sequence. But what has happened after the human genome project is that people said that we have to start making sense of other animals on the earth. So to April, this keeps on, the number keeps on changing, but there is a genome sequence site where you can go and you will find that to this date there are large number of sequences available for eukaryotes, prokaryotes, viruses, etc. So this was a huge project, and now this is increased. So how do you sequence the genome? So like I told you that because it is the same letter, you can use the same technologies to read the genome. That is why the genomics sciences has grown so much, because you did not need to have a learning of a different language. You could, if you are able to read a common language, then you can make sense out of it using the same sense of technology. So typically what you can do is you take any samples which can be from any part, and then you isolate the DNA. You have to find ways as to how you separate the cells and how do you take the nucleus and then how you take the DNA out of the nucleus. So there are lots of technologies now available, and then you can start to sequence them in different kinds of machines. So you can amplify them, you can see the DNA, you can actually see the RNA, then you start to understand how much is the quantity, how much is needed to sequence. I won't go into the detail, but there are a lot of technology advancements because of which from very small amount you can amplify the entire DNA into larger amount, and then you can put them through these instruments called the sequencing machines, which will read out all these genomes in definitive order, and then you have to have a huge amount of computational knowledge and computational resources to be able to synthesize the information out of these sequences. And pretty much this is what has been used to sequence a huge number of animals and plants, including humans on Earth. So now this was just a basic about, given we have got the letters. Now from the letters, how you get to answer these curiosities. For example, every genome now tells you one story. If you think about python, the python is a animal which can a snake is a snake which can actually feed an entire animal. Now, if you think, what would be the challenges of feeding an entire animal? You know, body organs have to increase massively to be able to engulf an animal sometimes which is very, very large in size. And then if you have that much of animal you are fed in, then you need to actually have a very good digestive capacity also to be able to assimilate that, right? So does its genome have something unique which allows you it to engulf a huge animal which is not present in other snakes, right? So when people sequence the genome, they could actually get certain informations as to, they have special kinds of genes which allow their organs to expand when they feed, or which allows them to metabolize better, as a result of which the food would not rot in its stomach and would get digested. Similarly, there are animals like oyster, right? They live in sea, and they have to engulf, or they have to take all the food material which is in the sea. If you are polluting the sea, they will have to take that in, but then how does it survive? It needs to survive itself. So it turns out that the oysters have got different kinds of survival mechanisms, so that even if it is exposed to certain toxins or certain kinds of things, and it would respond to those stresses through having these genes in place. So these are something which are called as the heat shock protein genes. These kinds of genes respond to stresses of different kinds. If humans have 17, they have 88 copies. So you can imagine that due to the niche in which they stay, they actually have evolution working in such a manner that it allows them to cope up with the survival. There are interesting questions as to watermelon, right? Grows in the desert. It has so much of water in it. How does it have it, right? So when they studied the genome, they found that they have a very uh, intricate mechanism of vascular system, which is because of highly evolved set of genes which could make such a kind of a vascular system. Then there are curiosities that there are certain animals which feed on insects, and it has actually got a very small genome because it is a carnivorous plant, meaning carnivorous insect, and it would plant and it would just feed on insect and take all its nitrogen and phosphorus requirement. So as a result of which, it can do away with a lot of other genes which are needed by other plants, because it can get all its energy resource by very efficient means. Then there are kind of microbes which can live in very highly radioactive environment. It's an extreme environment, and they can survive. So these are something which is called as Genococcus radiodurans, and people want to know how they do it, because if you have such intense radiation, then your DNA will tend to get damaged, and if DNA gets damaged fully, then the cells will die or they might not survive. So this is the kind of way by which you actually have these radiation therapy in cancer and all. But then there are also other kinds of problems associated with such kind of therapy, and one might need to know what is the way by which you can have efficient repair mechanism, and could we actually study these kinds of organisms which stay in very extreme environment and get to understand them. So when people sequence this, they found that they have a very efficient genome repair mechanism, because of which how much radioactivity you give it, they again come back, right? There are other interesting things. You think about elephant. Elephant has huge body size. They reproduce till a very long age. Their longevity is very high. But if they have such a huge size, then they would have larger number of cells, right? And it has been shown that if you have larger number of cells, that means the cells need to divide for quite a long time. So if the cells need to divide for a longer time, every division, they might start accumulating mutation, and that is what is the cause of many cancerous conditions. So then there is a paradox that elephants, why do they don't have cancer? Because that is what been seen, that even if you compare to human, elephants with that many number of cells have a very less cancer propensity. So when people sequence the genome, it turns out that it has actually mechanisms which could prevent from cancerous condition from happening, because of the copy of this gene which is called as TP53. There are 20 copies of these genes, and this allows it to kind of thwart cancer. There are interesting animals like naked mole rats, and this is otherwise it does not look very good, but these are burrowing mammals. And what is interesting about them is that people have found that they live for 28 years, and they don't have cancer, they don't also feel pain. So people want to know that why is it that these animals don't feel pain, they don't have cancer, because if their genome tells something, then some people can actually find out ways as to how this kind of an information would be useful to make anti-pain medications, or try to identify the genes which are responsible for causing pain. So, so these are some of the things which genomic people are trying to find. Now I'll try to get you to what Shreyas sees in these animals. So, when we sequence as genomic scientists, when we sequence genomes, we sequence it from a particular perspective. But what I learned from Shreyas is also a different way of looking at the same animals, even if the genomes have got sequenced. So, for example, I'll give you a few examples. For example, honeybee. Honeybee is a eusocial animal. It actually lives in societies, and he read that it actually has a very interesting mechanism of protecting itself by a heating mechanism. So if a predator comes to the nest, all of them come together and have make such a heat that they literally fry their predator, right? And then they have other kinds of things. They have to find nectar. They have to get that to make honey. And how do they go? How do they find the nectar? They should have very efficient mechanism to go to the right flowers to make the nectar. So many things people have sequenced the honeybee genome, and many things have come out, because you might know that there is a queen, there is a worker bee, there are drones. So they actually have a division of labor, and different members of that family do different kinds of work, right? So there are these worker bees, which they would actually first nurse the larva, and then they will go out and start foraging. So when is the time they will start foraging? There are certain genes which gets activated, which tells them that now they have to move out of the hive, and then they have to start forage for feeding back the other people in the nest. And so when they start foraging, they have to have different kinds of sensory system developed, because now they have to have a sense of direction to go for a particular flower, and then they come back to the nest, and also not forage on things which are very toxic to them. So many kinds of things have to develop in them. What is very interesting, and this is why I like this kind of an interaction with kids, is that once he told me about this social insect, I started reading up, because he asked me questions and I have to find that out from genome. Now, from his kind of emphasizing to read, I also got into some interesting reading. So I learned some very interesting thing about honeybee. For example, there was an article: honeybees can't practice social distancing, so they stay healthy in close quarters by working together. Now, isn't this very situation of today's pandemic, right? For honeybee, it's like every day is a pandemic situation, because they stay in groups. Their hives are extremely humid. The temperature is such that it can actually favor growth of a lot of pathogens and all. And they are sometimes in very close contact with each other. So anybody getting infected in their colony will actually make all the bees get infected very fast, or the brood getting infected very fast. So it turns out, over a period of evolution, honeybees have actually developed lots of practices which is more like what is called as a social immunity. So in a community, if there is a problem, and if every individual faces the problem on their own, then there is a lot of strain on resources and on the individual. But if together they can actually practice a group behavior, which allows them individually not to maybe be so careful, then that is what is called as a social immunity, right? So honeybee has evolved mechanism which allows them to see the advantage of living in societies, and also to do certain kinds of things which allow them to actually survive in such kind of conditions. So for example, their cuticle is made out of certain toxins which will thwart fungal growth on them. They would also keep on grooming their fellow members in the nest to be able to make them clean. They would remove the dead ones. So this is called as undertaking behavior. So they keep on cleaning their hives. They will also practice something which is called as a hygienic behavior. So if some brood gets infected, then they will have a way to find out how to kind of clean them. They practice. So what they find is that if they sense that their hives are getting infested with some fungus, then they start to detect that, and then they go out and collect resins which they coat in their wax, as a result of which that becomes antimicrobial and it kills the pathogens, or it actually does not allow them to thrive. So they have an aseptic hive, which is what we are now looking for. And then the honey they also know which plants have medicinal property, antimicrobial property, so they will get the nectar from them and use it in the honey. So as a result of which, when the brood has it, they are immune, meaning they are immune boosted. And then they also actually sometimes would also remove themselves out from the nest by also committing suicide if they think that their health also threatens the health of the whole society at large. You can see that if people were to understand the bee culture, then many things which we are fighting in the pandemic times, if we were to behave in a socially kind of responsible way, a social behavior, just like the bee does, then we might be able to reduce the pathogen or the burden of the disease quite successfully. Then there are other animals like turkey. It's a scavenger bird. It has to feed on dead animals. Now you can imagine if a dead decaying animal is fed, then how much it could be a strain on the stomach, right? How does the vulture do? So this was Shreyas's question, and then I went to the genome, because how do they do that? And then it turns out that they actually have detectors, smell detector genes, which are higher in number to detect decaying smell of flesh. And then they also have genes which can detoxify all the toxic things in their gut, as a result of which they are able to kind of even eat the dead animals. And then there are changes in genes which are related to immunity, so that also they can fight the germs which are there on the dead and decaying animals. What is more interesting is that he came out with certain animals, and I desperately tried to find whether people have sequenced the genome. And some are very still curious animals whose curiosity should actually prompt people to start sequencing genomes. And for example, frog fishes, they are called fishes, but they cannot swim, but they are good at camouflaging, and they prey using a worm-like organ on an antenna. So how do all these things evolve? So these are some very very curious questions, and some of you can actually, if you want to be biologist in future, you can actually start to ask this. So because all over the world people are now sequencing genomes, they think that millions of genomes can, if they are sequenced, then one might actually get to know mm, about different forms and functions and life, and as a result of which they could know more about birds, more about animals, more about ecosystem, etc. For example, these are from these projects itself, that herbivores, do they have the same taste receptors which are there in carnivores? Or why have pangolins lost all bitter receptors, though they are insect eaters? Or there are certain kinds of aquatic carnivores like whales or sea lions. They don't need to taste food because they engulf them. So it turns out that whatever habitat they have, they are there. These receptors or the taste thing, the genes which allow you to taste things differently, have evolved differently. So if your habitat is rich in sweet food, then you have to have a perception for that. And then many of us have a lot of bitter taste receptors too, because many toxins are bitter in nature. And if we are not able to sense bitterness in fruit, then we might end up ingesting things which are not good for us, right? Similarly, there are birds which cannot actually taste sweet, but then there are these hummingbirds. Whereas interesting part about this is that it has to flutter its wings, and it has to get a lot of energy, and the energy it can get from sugar. So how does it do if it has lost the taste of sugar? So there are other genes which have evolved in this, which allows them to do that. So like this, many many interesting kinds of questions one can answer, or one can ask, and one can go and find out in the genes. And these are also true in humans. For example, just a minute. For example, in the humans also, many of us like bitter chocolate and many of us like sweet chocolate. Some of us will not like karela and all the bitter food, right? It turns out that there is a genetic basis to it, and some people are extremely perceptive to bitter taste, so that they will start avoiding bitter food. As a result of which, many of the food which are very healthy and nutritious in nature, like all the cruciferous vegetable, they will tend to avoid, and then they will start having lots of things which are related to high calorie, and they might end up having diseases like obesity and all, right? This would also be true for all of you might be going to Subway or some place to have burgers, and everybody want to make it different, have different preferences for different things in a burger, and that is because all these have molecules which are sensed by our taste and olfaction ability, which have a genetic basis. So who likes more onions, who likes more tomatoes, who likes more meat, who likes more bread portion, all of them could be actually determined by how we perceive taste, and that is because of what is our genetic makeup. So all the way from how an organism is was to what kind of behavior we have, there could be many secrets in the genome, right? So this is all about genome, but then how do people use this information? So there are many interesting things people are now using, because now we know that there is a uniform language. So now can we actually use this technology to be able to even ask even interesting questions? So some scientists became a little ambitious, and they said that let us try to see whether we can actually find animals in the forest by using leeches. So as you know, leech feed on the blood, and so when they had ingested the blood, if you take the DNA out of the blood, and then if you have a way to match it to what kind of animals it would have come from by DNA sequences, then you would be able to make a call as to how many animals are there in a forest. So some scientists, what they did was they used this experiment. They left some leeches in the forest, and then they took it out, and then they took the blood out of it, and they could actually identify certain species from the blood, because it fed on them, that there are certain species which they thought were not there in the forest. They could actually hunt them out. Similarly, scientists have used these flesh-eating flies' guts in Malaysian jungles to be able to also prospect for animals which they thought that they were lost. There are people who are using even the museum specimens DNA to be able to identify what makes an animal go extinct. So this is a person called Stephen Schuster. He works on this kind of a new area called museomics, where he takes specimens of animals. So he sequenced a mammoth's genome, then he looked at Tasmanian wolf. So he's looking at animals which have become extinct recently. Is there certain signatures? So if you sequence animals, then you detect whether they are in the verge of extinction, and then can some conservation strategies be evolved based on these kinds of ideas, and whether that could save animals. So, using his methodology, he was actually able to help out certain zoo in California to increase the number of certain condors whose number had dwindled to 40. And using genome sequences, they found that why they are shrinking, and then they went about using that information to do captive breeding, and within a year, I think their numbers increased from 40 to 400. So one can also identify questions from this. There are other more very interesting questions. For example, some scientists wanted to ask that when did humans start wearing clothes? Because fiber is something whose or archaeological evidence is not there. So our ancestors are called as naked apes, but then we started wearing clothes. How do we know when did we start wearing clothes? So he came out with an interesting strategy. There are these lices, which have to obligatorily feed on a fiber, where if they are body lice, and they have to feed on the hair. So now body lice has evolved from the head lice many years back. So what he did was he sequenced the body lice and the head lice in different parts of the world, and then he came to the conclusion that the body lice has evolved from the head lice nearly 40,000 years back. So then he went to archaeological evidence and he found that needles were also found around that time. So the premise was that if head lice or body lice had to evolve, then there would be fibers in the body in which it could come and feed. And so if head lice and body lice have diverged 40,000 years back, maybe humans started wearing clothes 40,000 years back. So isn't it amazing? Without, if we did not know how the lice feeds, what is its niche, what time it actually diverged by looking at divergence in human genome sequences, we might not have been able to ask such kind of questions in today's date, right? So there are other interesting things. Human genome got sequenced, and then we found that we ourselves in our body we are human not because only ourselves, but we have 10 times more number of bacterial cells. They colonize different parts of our body, and different bacteria or colonize in different niches depending on what food sources are there. But people from different parts of the world have the same bacteria in same niches in the body. What has also been shown is that if you actually have certain diseases like obesity, that could be attributed to because you host different amount of same bacteria in certain parts of the body, and that could lead to disturbances in your body physiology, as a result of which you get different kinds of diseases. So now it is considered that you don't call human only because of human cells, but we have 10 times more number of bacterial cells. They come with their own genome. So if human have 20,000 genes, then these bacteria together can actually contribute to 200,000 more genes. As a result of which we are now, so the whole bacteria is also called bacterial microbiome, what is called as, it's actually called as another kind of a virtual organ. So people are now, after human genome sequencing, cataloging microbiome from different parts of our body and trying to understand that. There are other interesting things. There was so because some of you might be also students who like to read English literature, you must have read about Richard the Third, and there was something very contentious about where did Richard the Third, where is his where was he buried? So 2012, they actually they found a skeleton in a coffin in a parking lot or some place of a and they found that when that skeleton could have belonged to Richard the Third. But then how do they go about finding it? So they actually trace the lineage of Richard the Third to certain living members, and they sequenced it from the skeletal DNA and from the living members and tried to match them, and they could actually trace that possibly this skeleton belonged to Richard the Third. But then there were some pictures of Richard the Third which had some things about his eye color, hair color, and it seems there are genetic variations which can also predict that. What is the color of the eye? What is the color of hair? And etc. So they actually sequenced, after sequencing the genome, they could actually recreate this person's phenotype, and they could, with all the other kinds of evidence of dating, radioactive dating, archaeological bone structure, and the DNA, they could actually say that that belonged to Richard the Third. So this was actually labeled as closing 500 years of a missing persons case with genetic data. So this is also very interesting aspect one can do. And now coming to the last part, that what one can also do now is sequence the coronavirus, because this is a deadly virus which has actually shaken the entire world, and one now needs to know what is this virus doing, right? Whether it is of one kind or whether it is of different kind, whether it is evolving differently. So you must have heard in the newspaper RT-PCR sequences, different strains. So all these are because once you know the sequence of the coronavirus genome, then you can start to align with each other and then to know which place it is changing, and then you use it for diagnosing, for contact tracing, and also try to understand how you can use this information for making vaccines, etc. So there was one strain in China, and then after that it moved to different parts of the world. And at every part of the world, it could have accumulated one or the other changes, just like I mentioned. As a result of which, all around the world, you would have different kinds of strains from the parental strain by these series of mutations. And these kinds of strains can now be by sequence cataloged into different varieties. And one now can sequence and try to see which part of the world they would have come from. And then based on that, they could also trace the other contact members. So this is just a small video that now there are resources where all these information can be accumulated, and over a period of time you can actually start to see what is evolving in which part of the world and whether you are getting new strains. And as a result of which, people can now trace movements, track people, and have different kinds of things available. So this is the way by which genome sequencing has also been used very rigorously in understanding coronavirus. And finally, I'll try to leave with a thought that be curious. If you are curious, then you have a more interesting way of learning things, and that learning becomes very effortless. So this is what Jagriti understood when she worked in my lab, because she wanted to know how you can make learning as fun. So she went about studying it with a fellow colleague who works in neurosciences, and she understood that there is a system in our body called the mesolimbic system, which gets activated when a mind is curious. And this is the brain's reward circuitry. It gives a feeling of happiness. So if you have activated the mesolimbic system, then you feel rewarded. Now, if you make learning as a rewarding exercise, then you start to form better memories, as a result of which you have more curiosity to learn. And not only she learned this, she actually did this as an experiment. So she wrote two passages on gravity, and you can see from the title, this talks about gravitation, and here it talks about guaranteed weight loss without exercise, right? If I tell you, if I give you these two passages, which one would you more gravitate towards? You would more gravitate towards, okay, what is interesting written over there? But if you read carefully, all of it, what is given in very dry language of physics, is actually converted into a language which makes you curious. For example, it says get up and jump. Why do you land on the ground and not float into the air? We land on earth, simultaneously the earth is revolving around the sun. How? Why? Right? So it is the whole passage then starts to read more by I am trying to read a story. And this is what she did in that science reporter, because what she did was she wrote these passages and made her parents, her brother, and her friends read it and try to ask which one did you find learning gravity better. So this is what she wrote, that this contribution is a true and unbiased reflection of a 16-year-old on how in a short summer internship in learning and some insightful interaction can impact her life and career choices. Through experiment, we also found out that learning should be made fun for better impact, and hence learning to learn is a critical skill, right? So she ends her passage with, the next time you're taking a walk and you see beautiful trees, ask yourself why their branches are spread out the way they are. When you write on a piece of paper, think of the force enabling you to do so. When you look at a rainbow, find out how it forms, for it is this why that has led to progress both in science and as a species. It's very profound. It's totally her creation, written by a 16-year-old who just had a experience in curiosity in the lab, right? So that's what I would like to end with. Of course, there was another student who also wanted to know the outer world from animals. For example, if you have so there is an engineering field now which is called as biomimetics, where they are using animal shapes and animal size to make better machines. For example, Mercedes Benz made a particular model based on this box fish, which has a very less drag and moves very efficiently. Or there is this building which is made on basis of how termites keep their houses very cool without any air conditioning by the way they have made it. And this building in US actually is inspired by this termite, and there is no AC here. Or people are trying to use a mold like dictyostelium to find out what is the best traffic solution. So these are called as dictyostelium. You give them food sources, and they find out the most best route to those food sources. And apparently, the kind of routes they take actually, if you so this is a map of England, and somebody did this experiment where they put the food in various sources, and then they asked the dictyostelium to take its route, and it took the most efficient route. And when they found it, they found that these kind of routes they take can inspire better traffic management solutions. So people are using dictyostelium models to find out how you can decongest traffic and get the most efficient route. So these are some of the very interesting engineering, archaeological, very many different kinds of questions you can ask if you start to understand nature and the genome, and also all the things it creates out of the genome. So thank you for hearing out, and I would like to end now, and I can take questions. I cannot promise that I know everything, but I'll try to answer some curiosity questions. Thank you.
Thank you very much, Mitali madam, for this wonderful journey of genome. Actually, you have cleared so many facts which I was not aware of. I came to know several facts, and I think I should thanks to Jagriti and Shreyas also. And we should learn from the ma'am how she is learning even from the students also. So we have few questions which we have selected. One, Bhavya Bhutani, ma'am. His
Yeah.
question is our height also determined by our genomes?
Yes, so people are looking at the height. So what is very interesting is that height has a lot of heritability. So people, if they are tall parents, then the children tend to be tall, and this is what has been seen all over the place. And then so people thought that let us find out genes which determine height. It turns out that you require a large number of genes to actually participate in the height. So there is a genetic basis to it, but people have not found one single gene which could cause these differences in, or this heritability in height.
Okay. Madam, one Mr. Paras Kumar want to ask one question. Many people, as many of the people said that this coronavirus is human-made virus. So, is it possible to make artificial virus as a biological weapon?
So very much possible to make anything now. There is a area of synthetic biology. So this is what is very important, that with the genomics area there was also an allied development of ethics. So we what we can do good with something, we can also do bad with the same thing, right? So it is possible to engineer microbes because you know what toxins are there. So bio warfare is becoming a threat, and one has to find out also. So that's why also genomics is needed to be able to understand whether there is an impending problem from bio warfare, what it could be to diffuse it, right? So things should be used for better purposes, but unfortunately our mind also works in different ways. So sometimes we end up making things which are also uh, so this is the area which needs to be understood also.
Right, ma'am. I agree with you. One Nina Puja question is: does mental illness like negative thoughts or depressive thoughts are also under the control of genomics,
Yes,
negative
so
thoughts.
there are so it is that if anybody is exposed to any kind of stresses, we actually behave or react to those stresses in different manner, right? So there are signalings to the brain which makes us uh, behave. We are depressed. We have bipolar. Lots of things. So there would be predispositions, but most of the time it is a mix of genetic and environment which actually plays out more important role in precipitating such kind of diseases. So people have seen that there could
Please
be
take if
genetic
more
basis
questions are
to
there.
mental illness, but then the some of the time you also have the same variation, but it doesn't present with illness. So there are environmental component, and if you know the environmental component to those things, then you have better way of intervening. So uh, so these are complex diseases where there is a complex interplay of gene as well as environment.
Thank you, ma'am. One more question. One Simran Yadav question is, ma'am, if anyone gets infected with coronavirus, then will there be any change in genome of the persons? If so, what will be the changes? If say coronavirus infects some person,
Uh-huh,
will there
uh-huh,
be some
uh-huh.
changes in its genome also?
So most of the time, it is the response to the coronavirus which actually leads to the illness. So coronavirus would not get into our genetic material. I don't think so to be able to transmit it to the next generation in that manner. But there could be changes which are like the epigenetic changes also is possible. But what happens is coronavirus genome is in the cytoplasm. Most of our genetic material is in the nucleus, right? Any change which has to be transmitted has to be a change at the DNA level to be able to exert its
What
effect
is
in
this?
the next generation. So what coronavirus does is it actually
Please claim it.
hijacks the host machinery to its benefit and sometimes to the problem of the host, because many of the machineries are also needed for host physiology, right? So it could change the host physiology, but changing the host genome would require even more it has to be more even more smarter than it is now.
Acha, ma'am, one more question is there. You talked about if someone is vegetarian and the other is non-vegetarian, is this genetic? Is
No,
something
no, no,
genes have to play
no.
a role in it?
No, no, no. It's a behavior. It's what you actually get into. So many times vegetarian also can become non-vegetarian. Non-vegetarians can become vegetarian. So it is not that you have a tendency to be vegetarian. So that's if that is what you've asked. So
One Mr. Fiber Hacklin, his question is why it is taking too much time for making coronavirus vaccine.
any vaccine development takes time. So coronavirus is very new to human. We have to understand its biology to be able to. So whatever is happening at a very fast track. Normally, it takes 10 years to develop a vaccine. So because we are suffering, we think that it is taking long, but the vaccine has to. We have to understand the biology. We have to understand its. Where it should not actually end up harming us, right? So we have to have lots of safety in place. We have to do trials in people to be able to say that it is good for human use, right? So
What is
these
this?
take time.
Uh, I think there are some more questions. One question is, do you think we can reduce large number of cancer patients by removing CRISPRs and removing proton collage? What to college?
Prolonger. Prolonger.
So by this,
Proto-oncogenes.
yeah.
Okay.
So can we reduce the patient number of cancer patients?
Mm. So we have to understand that cancer is very, very complicated to understand. It is an interplay of uh, triggers which precipitate it, and then people have different kinds of cancer of different organ systems, and say a core set of genes also are there involved, but they cause many same gene does not actually cause different kinds of cancer in one person. So even if you think about all these CRISPR technologies and all, these are all getting attempted, and depending on
This is your
uh,
question.
how the technology evolves, we will have to see it. But there has to be a multi-pronged approach to cancer management. So diagnosis is there, appropriate treatment, who should get what kind of treatment, how to minimize the side effect, how to. So there are other kinds of things which are being attempted to make the change at the genetic level at this point of time is not so much as how to more understand it and better diagnose it and treat it.
Ma'am, there are more questions. So one Anupama Sharma question is intelligence inherited?
Anybody can become intelligent. You have
Yeah.
to use your
Are there some
uh,
genes also which carries from one generation to another for intelligence?
so everybody has all sets of genes, and people are all differently intelligent. Somebody can be very good at maths, the other person can be very good at creativity, somebody can be very good at tool making. So there are different kinds of intelligence. So I don't think that when you want to define intelligence, you have to define it in an appropriate manner.
Can someone, Madam, one Aditi Bhardwaj has a question. Can someone inherit family problems like hypertension, diabetes, controlled by lifestyle changes? Can someone inherit family problems like hypertension, diabetes?
Okay. As I understand, the question is that if somebody has inherited a, a problem which runs in the families, can a
Yes.
person control it? Right. So
Yes.
many of these diseases like hypertension, diabetes, cardiovascular disorder are manageable. Uh, so if it runs in the family, there is a chance that many members in the families will be impacted. But if a proper lifestyle or a proper exercise or dietary regimen is uh, carried out, then there is a possibility that you could delay the onset of the disease, or you could actually manage the progression of the disease. So all these complex diseases are an interplay of gene and environment, and if one understands which is playing out more, one can try to manage it by appropriate interventions. So, genetic variations is something which is not in your control, but if there is an environmental component to the disease, then that is very much in your control, and there could be many different ways. It could be change in diet, change in lifestyle, change in exercise. So, lots of options could be there. One needs to discover and find them.
My one of my colleague wants to ask one question.
Ma'am, it is a question related to Down syndrome children's.
Yeah.
So, is the gene responsible for Down syndrome is always has negative impact or some positive also? And the second part of the question is, in what field Down syndrome children can do better?
I am not qualified for this, and I would not be able to tell you because so there is trisomy, and there are now many rehabilitation programs, as I understand. But since I am really not delved into this area, I would not be able to make a comment.
Thank you very much for Madam such an enlightened lectures. I think our viewers are very much benefited by this lecture. I would request our viewers to please like this lecture and also subscribe to our channel so that we can come to know how your feedback. Thanks a lot, madam, for for taking out time from your busy schedule and for this wonderful online lectures. We would like to have your lecture again when our center is active and we have visitors at our centers. So we'll be waiting for that day also. I think things will improve soon. Thank you very much to all our
Thank you so
viewers.
much.
Thank you, ma'am.
Thank you so much.
Thank you very
Thank you
much
so
to
much.
all the schools who are listening to this wonderful lecture. And thanks all to my colleague also. This was our first time experiment for
Okay.
live streaming of the lectures. I think
Okay.
we had some problem, but majority of the program ran very well. So thanks to my team also. Thank you, Mitali ma'am, once again. Thanks to schools once again. So see you soon. Thank you. Thank you very
Thank
much.
you. Thank you. Thank you. Thank you.
Thank you, ma'am.